Welcome to another edition of AP Biology with Mr. Rickert. Woo, it's early. I need more coffee. Okay, let's talk about Unit 2. So this is, for many students, woo, need some water too. How are you all doing today? Hopefully you're not too stressed trying to learn all this material. One piece of advice I would give you. So one thing that helped me be successful in high school. So in high school, I was valedictorian. And one of the things that I think differentiated me from some of my peers who were also excellent learners, very good at memorizing things, very top academic students, was I didn't procrastinate. So I know some of you are really good at studying the night before for things. I personally, as a student athlete, I liked sleeping. So if I didn't get enough sleep, I would really feel it at practice the next day. So I always tried to get my work done early. So I would encourage you, especially for this class for AP Bio, try to get things done early. You'll be able to retain things for longer if you're not just cramming them the night before. That's essential for a long class like this that builds on itself. So keep that in mind. Okay. Okay, almost ready to start. But if you're watching this, you get the full uncut version. So hopefully that's fun for you. Let's look back at the most important document you have for this unit, the Unit 2 guide. So as we talked about functions of the different parts of the cell, we spent a lot of time on the cell membrane. And in 9th grade biology, or 10th grade for some of you, with the plasma membrane, we really just tell you it's a semi-permeable membrane, and make sure you understand that. So we quiz you, we test you on the fact that it's semi-permeable. But actually, there's much more to the membrane than that. As we've talked about in class, the membrane is not just composed of the phospholipids, which create this semi-permeable membrane, but also there's proteins, which allow for the transfer of certain things in and out, so embedded proteins. Also, we have steroids. So they want you to know for steroids, it seems like the only thing you really need to know is they're in there. Cholesterol is the steroids in eukaryotes. There's other steroids in other living things. Let's check out cholesterol real quick. So cholesterol is interesting. So if we do a search of cholesterol, you're going to see lots of things about, controlling your cholesterol levels, cholesterol and medication, etc. We're not really concerned about that. The concern with cholesterol, having too much cholesterol in your diet, is that it could build up in your bloodstream. However, that's not really what we're so worried about in terms of your cells. Cholesterol that's embedded into your cells is actually a good thing. As you see, it's a very large, mostly nonpolar molecule. It is a type of lipid. Not all steroids are lipids. However, cholesterol is. And so it's going to be integrated into the lipid bilayer very easily. It's going to interact with all of those parts. The only part that's tiny bit polar is this oxygen part. But because of resonance, the electrons in the oxygen, well, I guess it's a little polar, but the fact that we have this one very little polar part in this very giant molecule is not going to really affect its ability to interact with the cell membrane. In fact, it might even help because this might be able to interact with a little bit with the slightly polar oxygens in the phospholipid membrane as well. So overall, you can think of cholesterol as a non-polar lipid, mostly made out of carbons and hydrogens. So that's pretty much it. Yeah, and it's gonna allow for fluidity. Why does it allow for fluidity? I just know that it affects the interactions between the phospholipids. By separating out the phospholipids, it's gonna allow, it's gonna have important functions, allowing the cell not to get too rigid or too fluid. So let's see if there's anything else that Wikipedia could tell us. So cholesterol composes 30% of cell membranes. That's a lot. Did not realize it was that high. I wonder if that's by weight or what. It is required to build and maintain membranes and modulates fluidity. Yeah, that's what we've been saying. Oh, cool. So that's what I just said. I didn't know that. So the one polar part, the hydroxyl group here in red, this actually interacts with water molecules surrounding the membrane. So that's cool. So just like the phospholipid membrane has a polar part and a non-polar part, this slightly polar hydroxyl group is able to has actually an important function. It interacts with the water molecules. So this is on the edge of the membrane, and these are all embedded. Very cool. I did not know that. It's not in your book. So it increases membrane package, which affects fluidity and integrity. Okay, so it plays an important role in animals in particular, because we don't have cell walls, we need more rigidity. So probably there's less cholesterol in plant cells. That's good to know. Cholesterol can also have a role in intercellular transport, cell signaling, and nerve conduction. That's cool. This stuff seems probably, in my opinion, beyond the scope of an introductory biology class, but just be aware that it does have other functions. But the primary function that we're going to focus on in the class, and indeed is the only one mentioned in the AP guide for you to know on the AP test, is how it affects cell rigidity, which is something that is going to be different between animal cells and plant cells. So let's look real quick about cholesterol in plant cells. So that's interesting. Some prokaryotes actually have a different type of lipid bilayer. That's interesting. And that's important if they live in hostile environments. Yeah, so the amount of cholesterol in the cell membrane fits well with the particular needs of that membrane. So they don't get into it here, unfortunately, but an animal cell would need more cholesterol in order to have the needed rigidity, and a plant cell would need less cholesterol. They're both eukaryotic, so they'll both be very similar cell membranes, but the fact that this plant cell has the cell wall means it's going to be less necessary. Okay, well, that was some interesting research. Hopefully you found that informative. Let me leave that picture up, though. That's a good one. Okay. So now to the main thing that we wanted to talk about, which was osmoregulation. Okay, sorry for that delay. I was just reviewing the material one more time. So basically what we're going to talk about in this quick video is osmoregulation. So by the end of the video, you should have a clear understanding of what is isotonic, hypotonic, and hypotonic, and how that relates to osmoregulation. You also see these terms like low osmolarity, high osmolarity. Don't worry about those. I think in the context of the problem, you should be able to figure it out. But in your own writing, I prefer hypertonic, hypotonic, isotonic. I think those are the clearest terms. Those are the ones I'll be using. I'll fold in the other ones for clarity's sake, though. Okay, so here's the big idea, is that water potential is equal to psi, which is equal to the pressure potential on the outside of the cell, plus the solute potential, which we can get by calculating it. So let's talk a little bit about that and how that relates to osmolarity. Okay, so let's for a second ignore the fact that we're talking about cells. Let's just talk about diffusion for a second. So if you have a container of soda, and here's our lid, and you pop open the lid, what's going to happen? Well, it will maybe make a little bit hissy noise, but what if you shook it up a lot? What are you doing by that? Well, what you're doing when you shake up this Coke can, C for Coke, if you're shaking it up, you're increasing the internal pressure because you're taking the gases that were diffused into the water, and you're causing them to want to expand, create all this outward pressure. That's a type of environment where we have lots of pressure wanting to get out. And if, in that case, if you open it, it would fly all over the place. So anytime we have a fluid, whether it's Coke flying all over the place and squirting on the person's face because you pranked them and you shook it up before they opened their Coke that they were going to have at lunch, or whether it's gas flowing, it doesn't matter. As long as it's a fluid flowing from a high pressure or high concentration situation to a low pressure situation, this is what we're talking about when we're talking about diffusion. And we've all experienced that throughout our whole lives. So in a similar way, we can actually create water pressure in cells. So now let's talk about cells. We're just going to zoom in. So let's not pay attention to the food mosaic model right now. We'll have plenty of time to talk more about that in other videos, but let's just ignore it or just accept this is semi-permeable, go at some things in, some things out. Here we have the cell with all the organelles. And here, let's say we have pure distilled water. This is 100% water. Well, what do we know about 100% water? Well, it doesn't have anything else in it. It's just pure water. So this is what we would call a classically hypotonic environment. What does that mean? Well, it might sound like a good thing to surround your cell with water, but actually what you're doing by putting your cell surrounded by pure water is you're actually introducing a pressure gradient. Just like when you shook that soda can and it created pressure so that when it opened it burst, it created all that energy. In a similar way, actually by removing all the things that are naturally around the cell and putting it in pure water, just like we talked about what would happen to a cell, what would happen to human cells if you replaced IV fluid with pure distilled water, and how that would be detrimental. In the same way, this is detrimental. And the reason why is because you're actually creating a pressure gradient. So the pressure between the cell and the outside environment is equal to the pressure of, I think the subscript is P, yeah, P, of the external environment press. So this is the water potential between the outside and inside. Now this term is usually going to be negative. This term will be positive. If it's isotonic, these should cancel out. We have no pressure. But by putting this in a hypertonic environment, the cell still has some solutes. It still has solutes. So this is going to be a negative term, and this is going to be 0 because it's going to be decreased because it's just pure water. We don't have any solutes in there. So it's going to be equal to negative I C R T. Don't worry about the math so much, but just know this is going to be negative. The cell is going to be wanting to expand because this term is 0. You replaced all the things that are normally outside the cell, and it's now just pure water. Okay, so that's maybe not so good, especially in plants. This would be actually a good thing because they want to, there's not cell and then water, there's a whole bunch of cells. And so by filling all their vacuoles, they're actually going to be able to perform their functions better. But for most cells, we don't want to put them in this sort of environment where we're removing the normal solutes in the environment. So what if instead of that, we used IV fluid, which is what they give people when they're sick and in the hospital, and they need fluids in order to be healthy. So obviously, if that's what medical professionals are giving people, you might guess that that's actually good. But let's talk about why. So IV fluid has many solutes in it. And we won't get into the medicine. I invite you to do some research and find out exactly what they put in the IV fluid. But what it actually is a classic isotonic environment. Now, that doesn't mean it has the same exact things as your cells. What it does mean is it has the same concentration of things. So while your cell is filled with proteins and carbohydrates and lipids and things allowing it to perform its function, the IV fluid actually has the same concentration of solutes in it. So let's just let's just check for a second. What's a reasonable concentration for a cell? So what is the solute concentration of an IV solution? So 5% saline, that doesn't really answer my question. Okay, 0.3 molar. Okay, that answers my question. So yeah, they're listing lots of different things. That's not really helpful, but there's many different types of IV fluids and they have different things in it depending on their particular needs. But regardless, all of them are going to be about 3 molar. And if anybody finds a more accurate number, feel free to let me know, email me or message me, but it's going to be about 3 molar. The concentration of this fluid is going to be about 3 molar. That means about 3 molars of different solutes mixed into this mixture. That helps it to balance out the pressure due to the solutes in this cell. So there's not going to be flow of water in or out. Lastly, let's talk about what if you drink lots of salt water. You have probably heard you shouldn't drink salt water, but why is the case? Well, in this last example, salt water in the ocean is a classically hypertonic solution. And hyper, the way I remember it, is there's a lot going on. So there's a lot of salt in salt water. This is not really what the Greek means when we get into the actual meaning of tonicity, but it's an easy way to remember it and it'll serve you fine on the AP test. So hypertonic, there's lots of things in there. And so it's a hypertonic solution. So in the first case, remember, water was flowing into the cell. So make a prediction what's going to happen here. Well, as you all said, I'm sure, in this case, water is going to be flowing outside of the cell through those aquaporins and through a little bit through natural diffusion. A little bit of water is able to flow even without the aquaporins, but with the aquaporins, it'll be much facilitated. Okay, so in this situation, it's flowing out of the cell. Why is that? Well, now we've increased the concentration. And as we said, the water potential is proportional to concentration. So by increasing the concentration outside, we've increased this term. That's the only thing we're changing. We're not changing what's in the cell. The cell always has its organelles, its proteins. It's just trying to maintain homeostasis. So the cell is, this value is actually staying pretty much the same. The only thing we're changing by changing the environment is we're changing the water pressure of the outside. And that's causing water to flow in, stay in homeostasis, or flow out of the cell. And so by increasing this, by adding lots of concentration to the outside, we now have a hypertonic solution. So if you're drinking lots of salt water, your body, first of all, is going to have to get rid of all the salt, which is not so easy. And also, it'll actually affect you on a cellular level. That salt, if it gets into your bloodstream, will cause all your cells to shrivel, just like pouring salt on a snail. So your body will work really hard to try to remove that salt from your body. But that's why you can't hydrate yourself by drinking salt water. It will actually do more harm than good, even if you were dying of thirst, because of this factor. So that's how I would describe osmotic regulation. Hopefully that makes a lot of sense. You don't need to, memorize all the different terms, but I would really try to be able to describe in your own terms what is a hypertonic solution, what is an isotonic solution, and what is a hypotonic and hypertonic solution, and how those situations happen in real cells, in real life. So try to link this to the different things we talk about. We'll also talk about chemiosmosis, which is the flow of ions, which is a little bit different, but it'll bring back the same idea of there's a gradient. In that case, the gradient is not from the pressure of water. The gradient is actually from the pressure, is from an ion, an electrical buildup on one side of the cell membrane. But it's the same idea. There's a net flow in one direction. So just like shaking up the Coke can is building pressure, when we change the solutes in the outside of a cell, making it artificially higher or artificially lower than the cell, we also build up pressure. And in the third way, the final thing we're going to talk about in this chapter in another video is what happens when you build up an electrical charge on one side of the cell membrane. But we'll get to that next time. Hopefully you're looking forward to it. Have a great day, everyone. See you next time.